The overall goal of the following experiment is to differentiate murine embryonic stem cells into various primitive and definitive hematopoietic lineages and to characterize all ES progeny. Murine hematopoietic cell differentiation is achieved by placing ES cells onto a confluent, fluorescently labeled M-strawberry OP9 stromal cell layer. Each day of differentiation or co-culture represents the various stages of hematopoietic development. At the appropriate harvest time, differentiated ES cell progeny can be separated from the M-strawberry OP9 stromal cell line by cell sorting. This ES differentiation protocol recapitulates primitive and definitive hematopoiesis as observed in vivo and can be used to characterize hematopoietic defects associated with knockout ES cell lines without the use of embryos.
Hello, welcome to the lab of Dr. Judith Gasson at UCLA's Johnson Comprehensive Cancer Center. My name is Halisha Paz. The main advantage of the technique I'm going to show you today over existing ES OP9 co-culture methods is that this procedure allows for the passage of all day 5 and day 8 cells, as well as the complete separation of all ES-derived progeny from the OP9 stroma.
M-strawberry OP9 cells are grown in OP9 cell culture medium, which must be changed every 2 days. The OP9 cells should be subcultured 3 days prior to when they are needed for co-culturing. It is important to note that during maintenance, the OP9 cells should never be maintained at a density above 90% confluency. First wash the cells 2 times with PBS. Then add pre-warmed 0.1% trypsin EDTA for 5 minutes. When the cells have detached, add OP9 cell culture medium and transfer the cells to a 50 ml conical tube. Pellet cells at 300 G for 5 minutes at room temperature. After centrifugation, discard the supernatant and resuspend cells in fresh M-strawberry OP9 cell culture medium. Plate cells at a minimum density of 10 to the 4th cells per centimeter squared. On the day the co-culture experiment begins, the OP9 cells that were subcultured 3 days ago should be confluent. The ES cells should be cultured according to the standard protocol for your particular cell line. Wash ES cells 2 times with PBS. Trypsinize with 0.25% trypsin EDTA and incubate for 5 minutes at 37 degrees Celsius. Next, add co-culture differentiation media to the ES cells and transfer them to 15 ml conical tubes. Pellet the ES cells at 400 G for 5 to 7 minutes at room temperature. After removing the supernatant, resuspend ES cells in co-culture differentiation media. Use a 21-gauge blunt end needle to obtain a single cell suspension. Count cells using trypan blue and a hemocytometer. Now retrieve the flask of M-strawberry OP9 cells from the incubator. Remove media from the OP9 cells and add co-culture differentiation media to the flask. Plate 1 × 10 to the 3rd ES cells per centimeter squared to each flask of M-strawberry OP9 cells. Incubate cells at 37 degrees Celsius, 5% carbon dioxide with humidity. This is day 0, and the co-culture should look like this. Three days after the start of the ES M-strawberry OP9 co-culture, mostly large, flat clusters of ES-derived progeny with defined borders are observed, and some whorls of cells will have begun to form. Carefully remove medium from the co-culture and replace with fresh differentiation medium. 5 days after the start of the co-culture, proper differentiation of ES cells on a confluent layer of M-strawberry OP9 cells should result in hemangioblasts, which appear as piled-up whorls of cells.
The most critical aspect of this procedure is making sure you get proper differentiation on day 5 of co-culture. At this point, you could see the hemangioblast colonies not only microscopically, but macroscopically as well.
Carefully wash the co-culture 2 times with PBS and trypsinize cells with 0.25% trypsin EDTA. Wash cells off flask with ES differentiation media and transfer to a tube. Pellet cells at 400 G for 7 minutes at room temperature. After centrifugation, Remove the supernatant and add new media. Then use a 21-gauge blunt-ended needle to resuspend cells and to ensure a single-cell suspension. Count the cells using a hemocytometer. There should be a 100 to 150-fold increase in ES cell number from day 0. Reseed co-culture cells onto a new layer of confluent M. strawberry OP9 cells. On day 8 or 9 and day 12, count the cells again. Remove the media with the hematopoietic cells and reserve. Wash the monolayer one time with PBS to remove hematopoietic cells in suspension and reserve. Add 0.25% trypsin EDTA to flask. After trypsinizing adherent cells, wash cells off the flask with ES differentiation media and add to the hematopoietic cells found in suspension. Pellet cells at 400 G for 7 minutes at room temperature. Finally, resuspend the cells using a 21-gauge blunt-end needle and count the cells. The cells are now ready to be replated or used in downstream assays. The ES M. strawberry OP9 co-culture system can be used to model hematopoietic development. Outlined here is an overview of the co-culture time course. Days when differentiated ES progeny need to be transferred to new M. strawberry OP9 cells are indicated. Proper differentiation of ES cells on a confluent layer of M. strawberry OP9 cells will result in the production of FLIC1 positive hemangioblasts at day 5 of co-culture. Hemangioblasts are the common mesodermal precursor for the endothelial and hematopoietic lineages and should appear as piled-up whorls of cells as shown in this image. As the co-culture proceeds, 2 to 4-celled clusters should appear at day 6 or 7 and grow into larger cell clusters by day 8 or 9. Hematopoietic precursors are present by day 6, while terminally differentiated hematopoietic lineages appear by day 14. In contrast, a co-culture with improper ES cell differentiation will result in ES-derived progeny that do not appear in a whorl pattern at day 5. The absence of cells in a whorl pattern indicates improper hemangioblast formation. A co-culture with improper hemangioblast formation will ultimately result in stunted hematopoietic lineage differentiation and should be discarded. With this modified ES OP9 co-culture protocol, ES-derived progeny are M. strawberry negative and can be flow sorted from M. strawberry positive OP9 cells on any day of co-culture for evaluation. Results from a representative experiment are shown here. Previously published ES OP9 co-culture protocols have focused solely on the passage and evaluation of the non-adherent floating cells. In this modified protocol, all ES-derived cells, both adherent and non-adherent cells, are passaged and can be evaluated. This leads to increased yields of hematopoietic cells, allows for the identification of rare populations of cells, and provides for complete characterization of all ES progeny, both hematopoietic and non-hematopoietic.